Poor nutrition triggers an immediate, aggressive cellular crisis. Globally, the surge in midlife obesity is tightly bound to our pervasive consumption of ultra-processed, energy-dense fast food diets. Adipose tissue was long categorized as a passive storage site, a simple mechanical depot for excess calories. We now know that a fast food diet catalyzes meta-inflammation, a state of chronic, low-grade systemic inflammation. This state reconfigures the host's physiological landscape, requiring us to treat midlife obesity as an active immunological disease rather than a metabolic failure. Patients with diet-induced midlife obesity present a stark clinical paradox. The innate immune system, the body's first line of defense, becomes hyperreactive, operating in a constant state of trained aggression. Simultaneously, the adaptive immune system, responsible for targeted long-term memory, undergoes premature senescence and exhaustion. These opposing failures occur in tandem. We see heightened innate aggression on one side and adaptive exhaustion on the other. To understand how a single diet creates this bifurcated failure, we must deconstruct its immunometabolic pathophysiology. This dual failure is the primary mechanical driver, translating poor nutritional input into accelerated whole-body aging. This table identifies fast food's specific biochemical profile, combining high concentrations of saturated fatty acids and refined sugars with a critical lack of fiber. Saturated fatty acids like palmitic acid act as signaling molecules, directly binding to toll-like receptor 4 on immune cells. As the body attempts to store this massive caloric influx, the adipose tissue reaches its mechanical limit of expansion. The individual fat cells undergo a severe state of hypertrophy, expanding so rapidly and violently that they outpace their local oxygen supply. This localized hypoxia activates a specific protein known as hypoxia-inducible factor 1 alpha. Once activated, this factor induces the expression of chemokines that recruit monocytes straight from the bloodstream into the adipose tissue. Inside the hypoxic fat, these infiltrating cells become adipose tissue macrophages, shifting from a resting state into a pro-inflammatory M1 phenotype. These macrophages release cytokines like TNF-alpha and IL-6, which disrupt insulin signaling pathways systemically and initiate whole-body insulin resistance. Beyond the fat tissue, the fast food diet directly triggers the innate immune system's central intracellular sensors. Inside the cells, the NLRP3 inflammasome recognizes the sterile cholesterol crystals and saturated fats of a Western diet identically to a bacterial infection. This activation triggers the release of interleukin-1 beta, which signals the bone marrow to produce an oversupply of immune cells, specifically granulocytes and monocytes. This cycle establishes the state of trained immunity. The nutritional insult induces epigenetic reprogramming directly within bone marrow progenitor cells. Specific pro-inflammatory genes are left permanently unwound and accessible, primed for rapid transcription. This epigenetic shift leaves the innate immune system in a persistent alarm state, responding aggressively to minor stimuli after the initial dietary insult ceases. While the innate system hyperactivates, the adaptive immune system in midlife obesity undergoes accelerated decay. Diet-induced obesity drastically accelerates premature thymic involution, where functional tissue is replaced by encroaching adipose tissue. This fatty infiltration surrounding the thymus increases the cell death of developing T cells and reduces the pool of available lymphoid progenitors. The clinical result is a reduction in the output of new, naive T cells and a severely restricted T cell receptor repertoire. Chronic inflammation also leads to the functional exhaustion of existing T cells. This chart shows how memory T cells in obese subjects overexpress inhibitory receptors like PD-1 and BTLA. Single-cell RNA sequencing confirms these cells reach a state of exhaustion identical to that seen in chronic viral infections or cancer. This simultaneous thymic loss and T-cell exhaustion leaves the host immunodeficient, unable to recognize novel pathogens, while the innate system causes maximal internal damage. The systemic immune failure of midlife obesity manifests directly in the central nervous system. This connection is facilitated by the liver-brain axis. In non-alcoholic fatty liver disease, the lipid-filled liver continuously pumps pro-inflammatory cytokines into the bloodstream. Lipid accumulation in the liver creates a persistent cycle of inflammatory release that travels throughout the body. These specific cytokines cross the blood-brain barrier, entering the central nervous system through active transport mechanisms. Once inside, these cytokines activate microglia, the brain's resident immune cells. This activation drives neuroinflammation, promoting the formation of beta-amyloid plaques and disrupting synaptic plasticity in the hippocampus. Clinical data indicates that high-fat diets can induce measurable memory deficits in as little as 3 days, long before significant weight gain occurs. Midlife obesity is an independent driver of cognitive deterioration, linking dietary choices directly to an increased risk of dementia. The hyperactivation of the innate system and the decay of the adaptive system merge into the clinical concept of inflammaging. This crisis results in the accumulation of senescent cells, particularly CD8+ T cells, which leak a toxic secretory cloud into their environment. This is the senescence-associated secretory phenotype, or SASP. SASP reinforces systemic decay through the unregulated secretion of factors like IL-1 beta and TNF-alpha. Midlife obesity fast-tracks this dysfunctional profile, forcing biological age to outpace chronological age. This raises the obesity paradox regarding the reversibility of this systemic damage. Caloric restriction and anti-inflammatory diets can improve metabolic markers like fasting glucose and adiposity. However, weight loss does not immediately restore the status of exhausted T cells. The epigenetic training, established in the bone marrow, resists immediate dietary correction. Restoring homeostasis requires clinical strategies that target the entire gut-liver-brain axis to quiet the persistent cellular alarm. Because the fast food diet leaves a structural epigenetic memory that accelerates decay, immunological prevention in midlife is fundamentally superior to remediation.